I'd like to introduce our last speaker, Professor Eric Rubin, a physician scientist from the Harvard School of Public Health. I think one of the things I could say about Eric is that he's one of the few people in the world that can follow John.
No.
He's, for someone who says he doesn't believe in genetics, of course, that means he's one of the premier molecular geneticists of TB in the world. Eric is best known for work he did with his postdoc Chris Sassetti to knock out, by transposon mutagenesis, every gene in TB and measure the phenotypes under different conditions in culture and cells and animals. And it really changed the field. So there's a before Sassetti and Rubin and after Sassetti and Rubin sense to the TB field. Much more than that, of course, Eric's one of those people that galvanizes the field. He's got a tolerable sense of humor. He's a little bit shy about asking questions in meetings, but you know, he has, like many of the speakers we heard today, collaborations all over the world, and it's just full of 1,000 ideas. So instead of discussing his sort of oldies but goodies portfolio today on transposon mutagenesis of TB, he excitedly told me that he would like to talk about mycobacterial cell division. And I said, that's great. And then, of course, today he said, well, he changed his talk. So I don't know what he's going to talk about, but I know it's going to be great. Okay. Eric Rubin.
Thanks, Tom. Thank you so much for inviting me. And Tom's right. I'm going to, I decided to change the talk. And really, after hearing John speak about years worth of beautiful work, actually, I wanted to talk about some experiments we did yesterday. And that kind of changes the title a little. And I was thinking it's hard to come up with a title. It doesn't have a ring to it. So so I thought about other titles, but it turned out that Rafi stole that. And so maybe some sort of hybrid with breaking up was hard to do yesterday might work. Now, we've heard about TB, and I'm not going to reintroduce TB. We've heard about how bad a problem it is and how we'd all like to help millions of people who are sick and dying of the disease in the world. But face it, the real reason we're in it is for the glory. And in fact, there's a history of TB glory. Because there are several Nobel Prizes that have some connection with TB, including the first one, for the treatment of lupus vulgaris, which is really TB, an unusual disease of TB of the skin, which was actually given before the Nobel Prize given for the discovery of the bacillus. And then, arguably, one could say that Domac's award for the antibiotic Prontosil in 1939 was somewhat TB-related. Prontosil was a drug that actually didn't turn out to be a drug. It actually is a prodrug for sulfa drugs. It was converted to sulfanilamide, a drug that was already known. But it turns out they don't take away Nobel Prizes, which is lucky for Domac. But there are sulfa-related drugs that were shortly afterward used for TB. And in 1952, Waxman received it for a real TB drug, streptomycin. And most recently, as of today, as we're speaking, another TB-related Nobel Prize is being awarded. It's being awarded to Venki Ramakrishnan for being the brother of Lali Ramakrishnan, who was supposed to be speaking here. And I'm the last-minute fill-in, so I apologize for that. But maybe... so then, let's get on. What am I going to talk about? So what makes TB TB? So again, Tom referred before to Chris's work in the lab years ago, which was to try to define the set of genes it required for the fundamental process of bacterial growth. And he used a method which we called transposon site hybridization, or TRASH, which was used to define essential genes, using transposon mutagenesis that John referred to, and the genome sequence, and taking a transposon that basically hops into the chromosome, disrupting the adjacent genes, and using engineered promoters within that transposon to produce transcripts which marked the adjacent chromosomal region. And so we could map every single insertion in a large population. And the way it works, very briefly, you're not going to see it again, so don't worry about it too much, is that you get insertions in a very large library. Every single clone only contains a single insertion, but across the library there are insertions all over the place, and in this cartoon there are insertions in every gene, these gray boxes. There are multiple insertions, and under this growth condition none of these genes are required. But under this growth condition, this gene is essential for the bacterium to form a colony, and therefore this population doesn't contain any insertions in that gene. If we make these trash probes and fluorescently label them red or green, mix them together, hybridize them to a microarray, we can identify the genes which are differentially represented in the two populations and figure out which genes are important for growth. And Chris did an experiment that was not exactly that, but comparing the insertions in a pool of growing bacteria with total genomic DNA as a sort of stand-in for transposon insertions all over the chromosome, and found out that altogether, just to form a colony on a plate, the bacteria need about 600 total genes, and strikingly, a lot of those have unknown functions. And so it brought up the question of how do we investigate these genes? After all, by definition, these guys are essential. They can't, if you make mutations in them that knock out the function of the gene, the bacteria won't grow and there's nothing to study. So how are we going to figure out what these genes do? And that's what I'm going to talk about largely today, is approaches to systematically study these essential genes, and then the kind of surprising result that we got from it. And in the end, the surprising result is going to look a lot like John's. So it was a great, thank you for introducing my talk, John. I'm going to start with something that John already spoke about, which is using regulated expression to study gene function. In John's talk, he talked about catalase, CatG, and using a tetracycline inducible promoter to turn it on and change the amount of expression in any given population. Now, this is a wonderful way of studying essential genes. CatG is not essential, but studying essential genes as well, because in theory, in the absence of the inducer, in the absence of tetracycline or anhydrotetracycline, you'll see it called ATC on some of these slides, you should get no expression. When you add tetracycline in you can get various levels of expression of the gene, so you can set the level of expression that you want. And this looked like a great way of studying cells. And maybe I'm going to give you two brief examples of that, I'm going to skip all the biology and go right to the meat. The first one is a story on cell division, work done by Mike Chow and Eric Head, a former graduate student and a current graduate student in the lab. They were interested in how bacterial cell division occurs. And when you look at eukaryotic cells, they're incredibly complicated. They have to do all these things when they divide. They turn pretty colors. They turn all this yellow and blue. But they have a nuclear membrane that has to dissolve, and you have to separate out all the chromosomes, and you have to remake all the organelles and make sure the organelles get sorted out correctly. And bacteria look like little blobs. All they do is split. And it doesn't really look like much is going on until you look back at the figure that is similar to what John showed earlier, the bacterial cell wall, which is quite a complex structure. It's got many, many layers. The peptidoglycan layer is covalent, which is a big polymer of sugars cross-linked by peptides, is cross-linked to arabinogalactan, another carbohydrate layer, which is then cross-linked to the glycolic acids, which, because it's not an isoniazid slide, is a very small layer on this slide as compared to John's. But in fact, there's this huge layer of 70, 90 to 90 carbon molecules. So when you divide the cell, you have to divide each one of these layers, and that's complicated, and they all have to remain intact during that cell division, otherwise the bacteria lyse and die. And remember that the bacterium is essentially surrounded by one big molecule. So how do you coordinate all of that? And worse still, the enzymes which are involved in degrading and synthesizing new cell wall components are on the outside of the cell. So they're not so easy to control from the inside of the cell, and there has to be some sort of signaling mechanism that coordinates these. And Eric and Mike were concentrating on one of the inner layer, not the outer layer that we heard about before, but the inner layer, which is called peptidoglycan. And to make a very long story short, they started looking at a protein called RPFB, which had been identified to be important in regrowth of dormant cells, but which turns out to be a lysozyme-like molecule, which is involved in cleaving the bonds between the sugar residues in peptidoglycan. And they use that to identify another interacting protein called RIP-A, which we called RIP-A, which is a peptidase, which is involved in degrading the peptide crosslinks, which hold those glycosidic chains together. And RIP-A, in turn, interacts physically with penicillin binding protein called PBP1, which is an enzyme which has two active sites, one of which synthesizes the bonds between the sugars in peptidoglycan, and another which cross-links the peptides to each other. And in fact, these three enzymes have different activities. RPFB cleaves this bond, RIP-A cleaves that bond, and PBP1 synthesizes the same bond that RPFB cleaves, and it also synthesizes this cross-link here, which is adjacent to the bond that RIP-A cleaves. So these have both complementary, synergistic, and antagonistic activities and form a complex. So why do they form a complex? And one of our thoughts was that it might be because they are required to prevent uncoupled degradation of the cell wall, so that these enzymes, which are potentially dangerous, don't just go and lyse the cell. And our hypothesis was that therefore that RIP-A has to be regulated, that it has to always be sitting in a complex, and the absence of RIP-A or the addition of too much RIP-A is bad for the cell. So in work that I won't show you, we show that these enzymes work at the septum, and so we theorize that there's a complex that's formed at the septum, and that complex has to form for normal degradation and synthesis of new septal peptidoglycan. So in the absence of, in a depletion strain where RIP-A is removed, we should see unopposed synthesis without degradation. To test this, RIP-A was known to be, from Chris's screen, was known to be an essential protein, so we couldn't knock it out. So instead, we did the trick that John described. We replaced the native promoter here with a tetracycline-inducible promoter, so that this gene would only be expressed in the presence of tetracycline, but not in its absence. And indeed, we found the effect that we thought we'd get, which is that the cells don't grow in the absence of tetracycline, and as we add more tetracycline, we get more and more growth. In fact, the growth at high concentrations of tetracycline is normal. But when we looked at the tubes, it wasn't that simple. The bacteria didn't appear to grow. In other words, their optical density, our usual measure of bacterial growth, didn't increase. But there were small pellets at the bottom of the cell, of the tube. And what were those pellets? Those pellets were these things, and I don't think it projects that well. But these are the normal-looking cells in the presence of tetracycline. But in the absence of tetracycline, the cells form these long chains. And they're septated, so there are normal septa formed between the cells, but they fail to separate from each other. So it appears that in the absence of RIP-A, we get synthesis of the septum, but we never get its degradation, and the cells can't be released from each other. Now, what happens if we express too much RIP-A? Then we'd expect that the uncomplexed enzymes could go somewhere else and perhaps cause trouble. And to do that, we made a very similar construct where we took RIP-A with a tetracycline-inducible promoter and put it on an episomal plasmid. So now it's in multi-copy, so that we get more copies of it and overexpress it inducibly in the presence of tetracycline. And when we do that, we see cells swell up like this. And what happens to those swelled up cells? Well, this is really embarrassing, but I have a movie, and it's very hard to show a movie after John. We don't have the fancy microfluidics that we'd love to. We'd love to talk to John about doing those experiments. It's basically Scotch tape to hold the cells down to the slide, and it doesn't work that well. So if you have trouble with motion sickness, you may want to close your eyes right now. But I'm going to have you concentrate on just a couple of things here. The other thing is that the inducer doesn't spread well through the agar pads that we're using, so we don't get an even effect on cells like one would expect. But if you look at, say, the tip of this cell in the presence of tetracycline and follow it, nothing happens because it's going the wrong way. Let's see, now something will happen. There we go. If you follow that, you can see it sort of balloon up, and it's dividing, and now there are more balloons here, and if we last long enough, as they're sliding off, they explode. So what's going to happen to those cells that swell up is that they're going to explode. And so overexpression is toxic, underexpression is toxic. So the expression level has to be exactly right for this enzyme. One more example, and I wanted to show this because Sloan is in the audience, is ESX3, which is an alternate secretion system. It's similar to a very highly studied system called ESX1, which is depicted up here, which is critical for virulence. ESX3, however, in Chris's screen, appeared to be essential. This is an alternate protein secretion system, which bizarrely is essential. And Sloan, who's sitting right up there, now in Carolyn Bertozzi's lab, and Amira, a former postdoc in the lab, went to look for what this system did, and they made the same sort of construct. They replaced this is not in TB, but in Mycobacterium bovis BCG, the vaccine strain. They replaced the native promoter at this end of this long locus with a tetracycline-inducible promoter to see what would happen. Now remember, I told you that this locus is essential. And what we found was a little bit surprising, which is it didn't appear to be essential at all. The bacteria grew fine. And it was only when we put them into a low iron medium that they didn't grow well. But we think that that's probably wrong. And the reason we think that this is wrong is if you actually look at gene expression for these bacteria that are grown under high iron conditions, there's still some ESX3 expression. And John referred to the fact that this promoter is leaky. The leaky expression of this promoter is about the same as the native level, this is an experiment that Sloan did, is about the same as a native level of expression of these genes. So in other words, we can't titrate down the amount of protein to the level that we would need to do to prove that this is really essential. Now, this did allow us to figure out the biology of the system. In large part, the system's required to acquire iron, which was a somewhat surprising finding. But we couldn't really show the phenotype that we wanted under ordinary circumstances. So, in conclusion, this isn't my last slide, actually, but these are all old, these are varying studies, but the expression levels have to be just right. You know, there's this Papa bear and Mama bear and baby bear beds. You have to get right in the middle in order to be right for some genes, that there's a critical level, which makes it quite difficult experimentally to systematically look at genes. And in addition, some genes are difficult to suppress far enough down so that you get the actual phenotype of depletion. So regulating gene transcription has its problems. So we went looking for another way of studying these, and that's to target the proteins themselves. Now, this is an old idea, and in fact, some of the oldest conditional mutants are protein targeted in that temperature-sensitive alleles are proteins which aren't stable at high temperature and, generally, and therefore fall apart, and they're beautiful ways of studying protein function. But there are a couple problems with them. First of all, there's no systematic way of making these. And secondly, mycobacteria, in fact TB, not Mycobacterium smegmatis that we heard so much about, but TB really only grows at one temperature. There's one example of a temperature-sensitive gene where the bacteria have to be grown at 32 degrees. But growing TB at 32 degrees takes months and months and months and months. So it's not really a very practical way of studying things. In addition, we're interested in what happens in vivo during infection, and mice, we can't really adjust their temperature all that much. There are these old experiments where you stick mice in a refrigerator for months, but our animal people won't let us do them. So this is not a perfect way of doing it. So instead, we wondered if we could use conditional degradation of proteins to study this. And we, meaning all this work was done by Vidya and Jun in the lab. And their idea was, instead of using a temperature-sensitive allele, they would use a known protein degradation system. This degradation system is called the CLIP system. The CLIP protease has been really well studied in several bacteria, particularly E. coli and Bacillus. And this system degrades proteins completely. It consists of a protease, CLIP-P, and a recognition factor, which in E. coli, for the purposes that I'm going to talk about today, is called CLIP-X. It's an ATPase that unwinds fully folded proteins, feeds them into the protease, and the proteins get degraded. Now, one way that the CLIP system has of identifying proteins for degradation is this system called the tmRNA system. If, under conditions of amino acid starvation usually, a growing polypeptide chain is stalled on the ribosome, it just gets stuck there. Now you've got a stuck ribosome, a stuck messenger RNA, and this piece of protein stuck onto it. So how do you free that up? Well, what happens is another specialized RNA called a tmRNA, which is both a transfer RNA and a messenger RNA, comes along, displaces the messenger RNA, adds an alanine, and then encodes the addition of this short peptide here, and releases the peptide, the polypeptide from the ribosome. And now the ribosome is free to go on and make more protein. Now, the protein that got made is truncated. It's too short, and truncated proteins can potentially be toxic. So ClipX recognizes this SSRA signal, the tmRNA encoded peptide, brings it over to ClipP, which degrades it. So that seemed to be a nice way of making proteins. But the problem is, of course, if you add that sequence to the end of a protein, it's unstable. It's immediately degraded. In fact, for the most part, you can't see it at all because the half-life of this protein is very, very short. So how could we go about protecting it? And our idea was to destabilize it and inducibly remove a protecting group. And let me show you that here. So what we thought we'd do is take our target protein, add the SSRA signal, and then add more sequence downstream. The SSRA signal, it turns out, has to be at the very C-terminus of the protein. If it's not, then it's not recognized by CLIP. And between this protecting group, which could actually be any peptide, and the SSRA signal, we inserted a cleavage site which would be recognized specifically by a particular HIV protease. And this is really with tremendous help from Tom Albert, who suggested this strategy, and with help from Phyllis Conkey and Zovu Ulenga, who gave us the appropriate HIV clone to do it with. So when the protein gets cleaved, nothing happens. We switch slides. Well, when it gets cleaved, this should be degraded. Okay, so does it work? And all the experiments I'm going to show you today, much as John largely did, were done in the important pathogen Mycobacterium smegmatis, which is just a very convenient model. I won't show you any of the TB work which we're doing right now, but that's because these are yesterday's results, and I don't have any TB results from yesterday. Okay, to see if this works, we produced GFP, which was tagged with that destabilizing tag, a conditionally destabilizing tag, and conditionally expressed HIV protease elsewhere in the cell, so that it would be TET inducible. And what happens is what we hoped. You start out with green bacteria, you grow them for overnight, and after overnight we see lots of fluorescence in the absence of tetracycline. This is ATC, the inducer in hydrotetracycline. But with the addition of tetracycline, we see almost complete degradation of the GFP, and that is borne out by a Western blot, an anti-GFP Western, where you see lots of GFP in the absence of tetracycline and very little in the presence of tetracycline. We could do that again with another reporter gene, a gene which encodes resistance to the antibiotic zeacin. So we tagged zeacin with this tag, and I should point out, because it's going to become important in subsequent slides, that along with the tag, we add an epitope tag here, so we can follow the level of protein by Western, and again put the protease somewhere else on the chromosome. And what we see is that in the presence of tetracycline and the antibiotic, we no longer get growth of cells. So we degrade the resistance protein, we kill the cells in the presence of the antibiotic, and we get a dramatic shift in the MIC to zeacin. That is a much lower concentration of zeacin kills the cells. So in the absence of tetracycline, cells are resistant to about 25 micrograms per mil of drug, and that drops down below 1 microgram per mil. So that's nice, and it's great that we can degrade GFP, because we certainly want to get rid of all the GFP we can. But what we're really interested in is, can we use this in a systematic way to study putative essential genes in the chromosome? Now, in order to do that, first we got some help from Chris Sassetti, who's now in his own very successful lab, and Kenan Murphy, both out at UMass in Worcester, who have helped perfect a strategy first designed by Graham Hatfill of inserting sequences onto the chromosome at high frequency, which just allow us to construct these strains in a higher throughput manner. So this allows us to take a gene that we're interested in and stick that tag into its chromosomal location. Now, the big advantage of doing this is that this gene still has its normal promoter. So the level of expression of the gene is normal, and in fact, it's regulated in the same way as as it ordinarily would be. It just has this C-terminal addition to it. And for many genes can tolerate C-terminal additions, not all, but many can. So this is a much more physiologic system in a way than regulating the expression. Now, to test this, we decided to choose some known antibiotic targets. And John has introduced us to antibiotics and what appear to be the complexities of antibiotics that we thought we previously understood. But we thought this is simple. If you knock out an antibiotic target, you'll see a phenocopy. You'll see the same effect of the depletion as you will with the antibiotic. And so the idea was that we would incubate overnight, and the experiments I'm about to show you, we incubated overnight, and then we did a Western blot to see how much of the protein was left. And because the tag has a MYC epitope upstream of the cleavage site, this will tag all of the target genes. So all of what I'm going to show you are Westerns, all done with this anti-epitope antibody. So let's start with INHA because John introduced us to it already. It's the target of isoniazid, or the putative target of isoniazid, although John, I think, raised some questions about that. It's involved in mycolic acid synthesis, as John mentioned, and overnight it's degraded almost completely. So it looks pretty good. But it varied quite a bit. RPOB encodes the beta subunit is the beta subunit of RNA polymerase. It's the target of rifampin, and overnight it gets degraded, but not completely. Cas-A is another enzyme involved in production of mycolic acids, and after overnight incubation, it's degraded a little bit, but not all that much. I will tell you that if we wait longer, we can get more complete degradation of this protein. And the DNA gyrase subunit, gyre-A, which is a target of fluoroquinolone antibiotics required for DNA replication, is also incompletely degraded, but somewhere in between. On the other hand, alanine racemase, which is required to make D-alanine, which is needed to make synthesize a cell wall, it's pretty well degraded, and I'll show you that in a moment. And dihydrofolate reductase, the target of several antibiotics in Mycobacterium smegmatis, we can use trimethoprim, is also degraded more or less completely. And how completely is completely? Just to show you an example, a few examples of that. Using titrations to try to figure out how much degradation occurred, alanine racemase is degraded. There's less than 3% of the protein there, probably considerably less. And the same seems to be true for dihydrofolate reductase, but not so true for gyrase, which is present at maybe a 90% reduction in the amount of gyrase. This is after overnight incubation. And for some proteins, that degradation is very rapid. In the case of INHA, most of it's gone at 3 hours. Alanine racemase and dihydrofolate reductase, they are gone almost completely at after 3 hours of tetracycline treatment. So this can be, so in some cases it's rapid, in some cases it's complete, and in other cases it's slow and not so complete. And we don't know the basis for that variation, but we certainly have speculation on that we can talk about later. So why did we do the experiment in the first place? Because we wanted to see if the whole method worked. Yeah, we can knock down proteins, but what happens when we knock down the proteins? What is the phenotype of these bugs that are depleted for these extremely essential proteins? And the answer is that some knockdowns have substantial effects. If we knock down gyrase, even though we haven't knocked it down completely, we get a slower growth, though not dramatic. RPOB, we get slower growth, though not incredibly dramatic. INHA, we get a very profound drop in growth. We see, and I should say this is all done by optical density, not by plating yet. We don't have the results of plating, so we don't know if these bugs are alive or dead, but there's still bodies there anyway. On the other hand, Cas-A, which we couldn't knock down very well, really had very little effect. And dihydrofolate reductase and alanine racemase have a mild effect on growth. And this is yesterday's results, as I said, but we've seen almost, in some other experiments, we see almost no effect after this overnight depletion, particularly for dihydrofolate reductase, where there's very little effect. But there's a problem, as you might notice, because these, I already told you, are the proteins that are knocked down the best. And we know that we're getting better than 95%, probably better than 99% knockdown of these proteins, and yet the cells are still growing. So we started this experiment by saying that this is a control. We knock down known essential proteins, and we should see the cells die, but they don't. For some they do, but for some they really dramatically don't. So why? It could be that when we degrade, we actually are degrading the epitope tag, which sits at the C-terminus, and therefore the westerns aren't really representative of the amount of protein there. So to test that, we took a couple of the enzymes that were targeted and assayed them. So for example, if we target the if we degrade dihydrofolate reductase and then assay the cells to see what's left, we see this is a slightly complicated experiment to look at because we have to use a control. This is in a crude lysate, and we're assaying for NADPH consumption, and there are many NADPH utilizing enzymes. But this is in the non-knockdown strain, the no tetracycline knockdown strain, and this is the background for that experiment, which is using methotrexate to inhibit the endogenous enzyme. And you can see that there's a fair amount of enzyme activity. It's more than 100 whatever activity units those are. Whereas in the knockdown strain, we really don't see any measurable activity at all, where the background is almost identical to the knockdown. Alanine racemase is similar. Here we're using a control strain that doesn't make any protease, and the levels with and without tetracycline are very similar of enzyme activity, but in something that does make the protease in the presence of tetracycline, there's almost no measurable activity. So knockdowns are real. Thank you. It could be that the depletions are not specific, that there's some sort of off-target effect in much the way that John mentioned. To test this, we looked to see if the depleted strains were hypersensitive to the appropriate antibiotic. So we assumed that if you knock down dihydrofolate reductase, the bacteria should become hypersusceptible to treatment with trimethoprim, which targets dihydrofolate reductase. And indeed it does. We get a big shift in the MIC from 10 micrograms per mil to 0.4 micrograms per mil. And this is gene specific, so there's a shift for the alanine racemase depletion to the drug cycloserine, for DHFR to trimethoprim, and for gyrase to ciprofloxacin, but not for any of the uninvolved. So it appears that the knockdowns are specific, and they're really targeting the proteins that we think they're targeting. So what's going on? We know that knockdown occurs at varying extents, and that for each antibiotic target, it appears that there are different amounts of excess capacity. Some we can knock down a little bit, and we get a phenotype. But others that are perfectly good antibiotic targets are present at enormous excess, at 100-fold or more excess in the cell. Now, if you were choosing a drug target, if you were talking about at the contest earlier, if you chose a drug target that's present at 100,000-fold excess, the judges would have laughed you out of the room. But in fact, some of our perfectly good antibiotics seem to do that. And how does that work? We don't know. It could be that the inhibitors are just so potent that they're able to inhibit down to the level, 1,000-fold decrease, where you start to see a phenotype. Now, the inhibitors we're talking about have 70 to 80 nanomolar IC50s in vitro, which is about the same as most antibiotics. It's on the order of most antibiotics, so they don't seem to be particularly potent. Still, it's conceivable that they're able to get into the cell well and really cause a lot of inhibition there. It's also possible that they're not really inhibitors. They're really doing something else. And in the case of DNA gyrase, gyrase doesn't just inhibit, fluoroquinolones don't just inhibit gyrase. They actually inhibit the DNA ligase activity. And that we end up with broken DNA. And it's the breaking up of the DNA that probably kills the cell rather than inhibition of the enzyme activity. Or finally, and this gets back to what John was saying before, it could be that these drugs have multiple targets. Certainly, their primary target has to be the one we think it is. because for most of these drugs, you can produce mutants whose mutations map to those genes. However, it could be that there are secondary targets that are necessary to act synergistically for these compounds. And any of these could be true, all of them could be true for or any one of them could be true for any specific antibiotic. But I'm afraid it means that we don't really understand the antibiotics we have, which is kind of surprising. That's where I want to stop. I want to thank my collaborators again, not just Chris, Kenan, and Tom, but also Dirk Schnappinger and G at Cornell. And these are some of the members of the lab, which has had a lot of turnover. Most of them didn't do anything here, so I don't really thank them. We just haven't gotten the recombinant. We're working on that. So Dan asks why didn't we target the ribosome, ribosomal proteins, which are targets for aminoglycosides, and the answer is we'd like to. Actually, we tried to target every single known antibiotic target, and we got 6 of the 8 that we tried. So the other 2 have yet to be made.
But is it on? Yeah. Oh, I'm wired. Okay. But that raises a good point, which is that if you look at drugs that target ribosomes that just inhibit the ribosomes, they're not cytal. They're static.
That's true. That's true. And I think that's probably why Dan
corrupting the ribosomes.
Well, I'll let Dan answer that question.
They're static. Inhibition of ribosomes is static.
Streptomycin is cytal.
But it causes misreading.
Exactly.
Yeah, so it's corruption. It's not inhibition, it's corruption. So a knockdown might look more like a static antibiotic.
Yeah,
that's what you were saying. Okay.
So in fact, that's almost certainly true because I haven't talked about another project we're working on with CLIP. The CLIP protease is involved in protein quality control too. It's actually essential in these bugs. And when you knock it down, you become hypersusceptible to aminoglycosides.
But
Don't let him ask another question.
But that wasn't
I
actually
have both
my
you
question.
guys. What do you think? I forgot the name of the guy. Who's the fellow who suggested that all these antibiotics work by a common mechanism, and that's
J.J. Collins.
oxidative damage?
Yeah,
J.J.
Jim Collins.
Maybe you two should address that.
Well, no, I think
We've
these
tried
are-
scavengers, they don't do anything.
And he hasn't tried doing it anaerobically. But you guys can't do that, can you?
They're not happy anaerobic.
So let me fill in the rest of the audience. Jim Collins at BU has a model where there's a final common pathway by which all bactericidal antibiotics act, involving the production of free radicals. And it could be that many drugs work that way, but it's way downstream of the actual even if that's true, it's way downstream of where the antibiotic is binding and having its activity. So, and we don't know if it's playing a role here or not. We don't even know if they're bactericidal yet because we haven't measured CFU.
I actually had a different question, which is slow growth. In the cases where you did depletion, you still see continued growth. Is that slow growth per se at the individual cell level, or is that just an increased death? So that the net population growth is slow, not because individual cells are growing more slowly, but you have a lot of death going on there.
Man, if only we had microfluidics, we could answer that question.
Got to move to Lausanne.
So we don't know. We don't know. There could be balanced growth, and there absolutely could be balanced growth and death, and it's a really good thought, something that we'd like to look at. You're squinting, Tom. That's okay. You can ask me later. Email. Okay. I want to thank you all, and thanks for the really lovely symposium. It was great. Congratulations, Tom.
Oh. So, Eric. Yeah. You can't get off the hook that easily. Okay. So, if you knock down a protein 99-fold, why do you get a 4-fold change in MIC?
Well, the MIC doesn't necessarily scale exactly with protein concentration. In some cases, it seemed to, which is kind of surprising. But remember that there is some susceptibility at low concentration, so it doesn't go down to 0. It's not a scale that really goes to 0. It's a scale that goes down to the concentration at which bacteria are susceptible. So it's not really a linear scale. So I don't know if that explains it, but I could more confidently tell you that if you compare different degrees of knockdown, there should be variation. But I don't think the absolute number is necessarily exactly right. These are enzymes, and therefore there's a threshold effect to them too.
Okay.
I know you obviously don't believe that.
Well, that's why we're going to get drinking later, and I'll really find out the answer. So, at the end, I'd love to thank all the speakers for coming today. It was a spectacular demonstration for me of how basic research and new ideas can change the discovery of drugs. And so I'd like to thank all the speakers for coming. We also need to thank the people that actually put on the meeting. Aviva Lipkowitz, thank you for doing all the organization. Tamina Madden here. These people made sure everything ran smoothly and did everything from plane reservations to arranging the lunches and dinners and things like that. So really spectacular job. I'd like to also thank Mark Schlissel for his support and Jeff Owen for his vision of starting SEND. Sam Wheeler, again, for his continued support. And I'd like to urge all of you to mark your calendars for December 9th, 2010. That's the date of the third symposium. So thank you all for coming today. It was a spectacular day.